develop. These are the mesonephric duct, which is lateral to the mesonephros, the part that develops to form the kidney, and then paramesonephric duct, which lies more lateral. These two different ducts will form different structures of both male and female reproductive system. In the indifferent stage, we have said there's condensation of the intermediate mesoderm cells and then epithelial thickening. This epithelial thickening eventually form the mesonephric duct, but the epithelial thickening around the gonadal ridge is very important in the formation of the cells of the gonads. When I talk about gonads here, I'm talking about both ovaries and the testes. We also have, lateral to this mesonephric duct, some invagination of coelomic epithelia. And this invagination usually continues on as a duct and grows parallel to the mesonephric duct. Its growth is guided by the mesonephric duct as it grows towards the cloaca. When the paramesonephric duct approach the cloaca, the two ducts come to lie close together and open at the cloaca, into the cloaca. Now, that is the two ducts we've talked about, which are very important, and we are going to look at what happens to this duct. We have also the first duct, the mesonephric duct, developed in the 4th week, and then the paramesonephric duct developed in the 6th week. And like we have said, between the 4th week up to the 7th week is where we have the indifferent stage of the gonads. So what happens during this stage? We have epithelia, the coelomic epithelia cells, which will have condensed over the mesonephric, over the gonadal ridge. Coelomic epithelia cells invaginate into the intermediate mesoderm. As they invaginate into the intermediate mesoderm, they form what we call germ codes or sex codes. This invagination into the intermediate mesoderm at the gonadal ridge is induced by chemical substances released by these mesenchymal cells in the intermediate mesoderm at the gonadal ridge, very close. Usually, and as they grow into the gonadal ridge, we also have the genes playing a role in terms of the actual development of the testes. At the different stage, like I've said, we do not have a specific structure of both male and female. Both of them are this. We have the epithelial cells, the coelomic epithelial cells, growing, forming germinal codes or sex codes, both in the male and the female. And then we have the germ cells from the yolk sac. These germ cells are the oogonia and the spermatogonia. These are germ cells that are for the males as spermatogonia, and the oogonia that are for the female. These migrate into the gonadal ridge, and come to lie close to the cells of the germinal codes or sex codes. These sex codes, as they develop, they go into the gonadal ridge. Because of the proliferation of the cells, these bulge towards the lumen. They bulge towards the lumen, forming a gonadal ridge. And as they continue proliferating, they will then be held to the posterior abdominal wall by a thin connective tissue element that we now refer to as gonadal mesentery. That gonadal mesentery. The germ cells, that is the spermatogonia and the oogonia, come to lie close to the sex codes. These sex codes cells surround these germ cells. The germ cells undergo proliferation. They undergo proliferation, they multiply within the gonadal ridge. And as they multiply, they are surrounded and incorporated into the epithelial codes, which were talked about as epithelial codes developing from the coelomic epithelia that invaginate into the gonadal ridge. So those epithelial codes surround the germ cells and now form tubular structures. Those tubular structures is what you're calling the seminiferous tubule. Seminiferous tubule. So within the seminiferous tubule, we have the germ codes or germ cells. And amongst those germ cells, we now have the sex code cells developing from the coelomic epithelia. These sex code cells developing from the coelomic epithelia will form the Sertoli cells. The Sertoli cells. So they'll form Sertoli cells. And then the mesenchyme tissue, mesenchyme cells, which were between these sex code cells, will differentiate to form the Sertoli cells. Now, what is the stage in which we now have these cells differentiating? What causes their differentiation? We have the testicular determining factor, which is a region within the gene, SRY gene, in the Y chromosome. And this testicular determining factor is what now helps in the secretion of formation of the Sertoli cells. So those germ cells surrounded by the sex codes, the sex code cells differentiate to form the Sertoli cells. They form the Sertoli cells. At that stage, that is now the beginning of the differentiation between male and female gonads. Sertoli cells will produce the hormone, anti-Mullerian hormone, and that anti-Mullerian hormone will inhibit further development of the Mullerian duct. The Mullerian duct is the one which is important in the formation of the oviduct and the uterus. So it inhibits further development of the Mullerian duct. And therefore, that is now going towards a male phenotype. In the 8th week, remember we talked about the 5th to 7th week as a different stage. In the 8th week, we also have the mesenchyme cells, again, under the influence of the hormones, differentiating from the mesenchyme tissue within the gonadal ridge to form Leydig cells. So those Leydig cells begin secreting testosterone. And testosterone is very important in the development of the mesonephric duct. So testosterone promotes development of the mesonephric duct, whereas Sertoli cell secretions of anti-Mullerian hormone inhibit the development of the Mullerian duct. So testosterone promotes mesonephric duct development. So those ducts in the male, we are going to see what they become. So at that stage now, you can see very clearly that we will have both male phenotype and female phenotype. Now, the germ cells, that is the spermatogonia, which migrate both to the cortex and the medulla, at that stage, they migrate into the gonadal ridge that is eventually forms the testes. However, those cells that are within the medulla are the ones that develop. The ones in the cortex degenerate. So any of the germ cells in the cortex will degenerate. The ones in the medulla will develop to form now our testes. So that's the difference. In the female, on the other hand, the germ cells, that is the oogonia, in the medulla degenerate. The oogonia in the cortex will be developing to form the oocytes. So it's opposite, and that's because of the different hormones. So they say by default, in the absence of anti-Mullerian hormone, we now have a female phenotype, and in the presence of anti-Mullerian hormone, you have a male phenotype. So in different stage, like I've said, you cannot differentiate between the two of them because the germ cells and the sex codes are all mixed up together. So that's what we have as far as male development is concerned. So the germ cells at the medulla, the germ cells usually will be together mixed up with the testicular codes. We are calling them testicular codes, otherwise it will be called germinal codes or sex codes, both in the male and female, but to be specific in the males, we are calling them testicular codes, those from the coelomic epithelia. Now, the mesenchyme tissue for the formation of the testes, as it develops, as the cells proliferate, it sort of detaches, but it's attached to the posterior abdominal wall by the mesentery, gonadal mesentery. The mesenchyme tissue just below the epithelia are induced now to form blood vessels and also connective tissue that forms the tunica albuginea. Tunica albuginea, which is like a capsule. But remember, this developing testis is covered by coelomic epithelia, so it will eventually be covered by a mesothelium comprising, it's covered by a serous fluid secreting membrane. That's what you're calling mesothelium. So note here the differences between male and female. Note also the closeness to the ducts here, both the paramesonephric duct and mesonephric duct. And paramesonephric duct and mesonephric duct are very close to the nephrotom, which is going to develop to form the urinary system. And also the migration of the germ cells from the yolk sac. So the germ cells originate from the yolk sac, migrate in the ventromesentery, and eventually come to the gonadal ridge. This migration is because of chemotactic activity. Chemical substances produced within the gonadal ridge stimulate migration of the germ cells towards the gonadal ridge. For mesonephric and paramesonephric duct, I think we talked about them. And the mesonephric duct develops in the 4th week as a thickened area within the gonadal ridge. Just lateral to the mesonephros. So this forms a mesonephric duct because it canalizes. This epithelial thickening canalizes, and the mesonephric duct in the male develop under the influence of testosterone. Remember, testosterone promotes development of the mesonephric duct. And testosterone is produced by the Leydig cells that will have differentiated from the mesenchyme cells within the gonadal ridge. So the fate of this mesonephric duct, it forms the epididymis, the vas deferens, ejaculatory duct, and seminal-vesicle in the males. Epididymis, vas deferens, and seminal-vesicle. During the development of the kidney, the proximal part of the mesonephric duct will have some tubular structures extending to the mesonephros. These thin tubular structures are what we call mesonephric tubules. Mesonephric tubules. The mesonephric tubules connect to the proximal part of the mesonephric duct. That is during the development of the kidney. The mesonephric duct also plays a role in the early development of the kidney. So during that stage, these mesonephric tubules now differentiate in the case of the males to form the efferent duct. The efferent duct. So that's what we have as the mesonephric duct. Now the paramesonephric duct developed in the 6th week. Again, this is during that period of the indifferent stage. And the paramesonephric duct developed as invagination of coelomic epithelia. It invaginates into the gonadal mesenchyme tissue within the gonadal ridge. This invagination as a solid coat of coelomic epithelia eventually canalizes. It opens at the center. So it canalizes and keeps its contact with the coelomic epithelia. So it's continuously open to the coelomic epithelia. Paramesonephric duct grows lateral to the mesonephric duct and is guided in its growth by the mesonephric duct towards the cloaca. As it approaches the cloaca, the two tubes come to lie close together as they open towards the cloaca. The paramesonephric duct now develops to form, in the case of the females, the oviduct, the uterus, and the upper part of the vagina. Oviduct, uterus, and the upper part of the vagina. The oviduct is open into the abdominal cavity because the coelomic epithelia was an invagination from the epithelial lining of the future abdominal cavity. That's why it makes contact. So the oviduct is open to the abdominal cavity by this embryological development. So we'll be looking at the female and we'll be looking at how do these structures develop. Now we have the descent of the testis. The testis originates within the lumbar region. It originates within the lumbar region, but comes to lie in the scrotal sac. The scrotal sac develops as labioscrotal swelling, and this scrotal sac now is where you will have the testis descending into and passing through the inguinal canal. So by the 3rd month of fetal life, it reaches the iliac fossa. By the 7th month, it traverses the inguinal canal, and the 8th month, it enters the external inguinal ring. So at birth, about the 9th month, it descends into the scrotum, or scrotal sac. And this is guided by the gubernaculum testis. You will also have a homologous structure in the case of the females, where we have gubernaculum ovary. And the gubernaculum ovary attaches to the labia majora. The labia majora is homologous to the scrotum. It's homologous to the scrotum. And that is attached to the ovary also. So we have also a descent of the ovary into the pelvic cavity. What happens if you have problems of descent? What happens if that gonadal ridge does not develop because of failure of testicular determining factor and also the homeobox genes? One, you can have an ectopic testis. Ectopic testis is where you have a testis in any other part of the body. Any other part of the pelvic cavity, it could be in the lumbar region, it could be within the abdominal cavity, that's an ectopic testis. But you can also have several enlargements of the gonadal ridge, several enlargements forming some testicular structures that are small. And therefore we call this polyorchidism. Polyorchidism will have more than one, you usually have a paired testis, but you can have more testes separated, but small testes, because within the gonadal ridge, there was a split, and therefore each of those developed into a separate testis. We could have cryptorchidism. In cryptorchidism, this is failure of the descent of the testis into the scrotal sac. So the testis could be within the inguinal canal, it could be in the abdominal cavity, and because of that, normally spermatogenesis takes place at lower than the normal body temperature. So with a cryptorchid, the spermatozoa will not be formed, and therefore the person is infertile. So that's what we have as a factor, because the temperature in the abdomen is higher than is normal body temperature, whereas spermatogenesis takes place at lower than body temperature, 2 to 3 degrees lower than body temperature. So that testis provides that type of an environment. You can also have some underdevelopment of the testis. Underdevelopment of the testis, again, because of hormones, it could also because of the chromosome aberrations. So that underdevelopment of the testis we call hypoplasia, and this is Klinefelter syndrome, as seen in Klinefelter syndrome. So you have very small testes. Anarchism, anarchia is where you have no development of the testis. So instead of having paired testes, you might have a single. It could be bilateral or unilateral. Bilateral means both of them don't develop. Unilateral means only one side develops, one side fails to develop. This because of failure of the induction within the gonadal ridge by the genes responsible for the development. And because sometimes you might not have adequate production of anti-Mullerian hormone, you might have persistent molar or paramesonephric duct. Persistent Mullerian duct or paramesonephric duct. So with this persistent paramesonephric duct, that is also another condition, sometimes we call hermaphrodite. Hermaphrodite, where you can have both male and female genitalia. It's ambiguous type of genitalia. So these are some of the congenital malformations that you can have with the testis. So usually we can have chromosomal sex determination. Chromosomal sex is determined at fertilization. Is it the X and Y chromosomes? It is divided by male and female. But you can also have gonadal sex. If it is Y, then you have chromosome, presence of Y chromosome. Then you have male testis, absence of Y chromosome. You have the ovary developing. Remember here we have testicular determining factor, the SRY gene on the Y chromosome that is responsible for stimulation of the development of the testes. Then we have the somatic sex. Somatic sex, that is phenotype, phenotypic expression. Phenotypic expression. How do you, what genitalia develop? That one is determined by the secretions from the Sertoli cells, the anti-Mullerian hormone, suppressing the Mullerian duct, and therefore suppressing the development of the oviduct, uterus, and vagina. Oviduct, uterus, and vagina. And just by looking at this, when you have no anti-Mullerian hormone produced, then you can end up with a uterus, you can end up with a vagina, you can end up with an oviduct. Or if you have less anti-Mullerian hormone being produced by the Sertoli cells. Then Leydig cells also determine phenotypically what develops. Remember, Leydig cells, we said, are very important in the development of the mesonephric duct. So that mesonephric duct eventually develops to form the epididymis, the vas deferens, we have talked about it, the ejaculatory duct, and also the seminal vesicle. So all this is phenotypic expression because of the hormones. You can notice here, hormones are very important in that determination. Apart from this, the other development of the external genitalia is also because under the influence of hormones, testosterone playing a very important role. Now, the cloaca is a common area. I've talked about cloaca. Cloaca is a common area where we have the ducts from the urinary system and also duct from the reproductive system opening into. So we have talked about the mesonephric duct opening into the cloaca. We have talked about the paramesonephric duct opening into the cloaca. We also have the digestive system opening into the cloaca. And if you remember when we were talking about the digestive system, we talked of urorectal septum. Urorectal septum dividing the cloaca into anorectal sinus and urogenital sinus. The cloacal membrane will therefore be divided into the anal membrane and also definitive urogenital sinus membrane. So what is the fate of this cloaca? So this is a common area from reproductive system, urinary system, and also the reproductive system. All of them open into the cloaca. Extending from that cloaca, we have extension to the allantois through the allantoic duct or the urachus, the allantois duct. So this extends at the umbilical region. Because of that umbilical region, that's where we have the allantois going, extending to the bladder, into the placenta. So what happens to this urogenital sinus? Talked about urorectal septum, if you remember. Primitive urogenital sinus. We said primitive because this is in contact to the allantois, but the primitive urogenital sinus eventually forms what you call definitive urogenital sinus. The upper part of this primitive urogenital sinus forms the urinary bladder, and the lower parts of this forms definitive urogenital sinus, and this forms part of the urethra and also part of the vagina. The lower part of the vagina will be formed from this. This is the phallic part that eventually is going to develop to form the urethra And from this, the phallus will develop to form the penis and the clitoris in the case of the males. This upper part will form the urinary bladder, and then the lower parts here of the paramesonephric duct. What was the paramesonephric duct? The endoderm will form, in the case of the males, is going to form the prostate. The endodermal thickening will form the prostate. Again, when you are talking about the digestive system last time, we talked about failure of the urorectal septum to completely fuse with the cloacal membrane. So if there's a failure, there'll be connection between the rectum and any structures that develop from the urogenital sinus. Some of these structures we have mentioned. You have the bladder, you have the vagina, and there could be connection between any of these that we refer to as a fistula. Rectovesical fistula between the rectum and the urinary bladder. You can have a fistula also in the vagina because the lower part, the part of the urogenital sinus develops into the lower part of the vagina. So the pelvic part of the definitive urogenital sinus developed from the prostate and membranous urethra. Prostatic and membranous urethra, also the prostate and bulbourethral glands. That is the pelvic part of the urogenital sinus. Definitive urogenital sinus. The phallic part will form the spongy urethra. And this spongy urethra, usually you can have an opening, the urethral folds. That urethral fold eventually fuses ventrally. And sometimes you might have a problem with the urethral fold where there is failure of fusion. If there is a failure of fusion of this urethral fold, then you'll have what we refer to as the epispadia, if it opens on the dorsal part of the penis, or hypospadia, if it opens on the lower part of the penis. Again, remember, these are congenital malformations arising from the definitive urogenital sinus developing. So you have a urethral vestibule, and then from this also you have a vagination of these endodermal cells to form the glands of Litre, which are in the penis. We are going to look at the penis, and we'll be looking at the glands of Litre that are mucus secreting glands. Just a recap, the mesonephric duct, also called Wolffian duct, develops to form epididymis, vas deferens, seminovesical, ejaculatory duct. And then the Mullerian duct, we have talked about what it forms. So let's now look at the male reproductive system. Let's look at the testis, look at the structure of the testis. But before we do that, I'd like to invite any questions. If you have any areas to be clarified, please put in the chat. Okay, looks clear. Now, the testis is an organ. It's both, we consider it both as an exocrine organ and an endocrine organ. The exocrine organ component is secretion of the spermatozoa. Secretion of the spermatozoa. In what we were calling before cytokine mechanism of secretion. cytokine mechanism of secretion. Then we have the production of spermatozoa. That's the endocrine part. There's production of spermatozoa in the process we call spermatogenesis. And also spermiogenesis. Then we have the storage of that spermatozoa until it's ejected out during copulation. It also plays a role in the production of hormones, specifically testosterone, and also other hormones produced by the Sertoli cell. Testosterone is produced by the Leydig cell, also called interstitial cell. And then the other hormones I've mentioned, I'll repeat mentioning, is by the Sertoli cell. If you remember, when you are doing endocrine system, we said the hypophysis produce follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone and luteinizing hormone. Under the influence of gonadotropin-releasing hormone from the hypothalamus. The parvocellular nuclei of the hypothalamus. So gonadotropin-releasing hormones stimulate the gonadotropes within the hypothalamus to produce follicle-stimulating hormone. Follicle-stimulating hormone stimulates Sertoli cells within the testis. It stimulates Sertoli cells within the testis, and Sertoli cells produce the hormones we have talked about. During development, it produces anti-Mullerian hormone, and then it also produces inhibin. Inhibin is an inhibiting hormone which inhibits secretion of follicle-stimulating hormone from the hypophysis. So it's a negative feedback mechanism. It also produces a hormone we refer to as activin. Activin stimulates the release of follicle-stimulating hormone from the hypophysis. So another feedback mechanism. It also produces a hormone we refer to as transferrin. And transferrin is very important in the absorption of ions by the developing spermatogenic cells. Absorption of ions by the developing spermatogenic cells. So you can see follicle-stimulating hormones stimulating Sertoli cells. Luteinizing hormone stimulates the interstitial cells, that is Leydig cells. And Leydig cells produce testosterone that are very important in the formation of the spermatozoa, that is spermatogenesis. On the other hand, Sertoli cells also produce androgen binding protein. The androgen binding protein is very important in the promotion of the testosterone activity within the developing spermatogenic cells. So you can see that Sertoli cells also play a role in that formation of the spermatogenic cells. The other role of the reproductive system is copulation, and this is the penis is very important in copulation. And we'll be talking about erection and what have you later on. So what is the structure of this testis? We're going to look at the testis as an organ. Every organ is covered by a connective tissue membrane, a connective tissue that we refer to as a capsule. So the testis is covered by a capsule we refer to as tunica albuginea. Tunica albuginea, a highly vascularized tissue. Tunica albuginea. This tunica albuginea that cover the testis penetrate into the testis as septa. If you remember when we were talking about before, we had a capsule penetrating either a septa or trabecula. So the tunica albuginea penetrate into the testis as septa, dividing the testis into lobules. Dividing the testis into lobules. The tunica albuginea or the septa converge at what we call the mediastinum of the testis. The mediastinum of the testis. And this is the area, highly vascularized area in the mediastinum, containing the rete testis, which we'll be looking at. During the migration of the testis from the lumbar region towards the scrotal sac, it carries along the blood vessels, nerves, and also is covered by the walls of the abdominal cavity. So it will be covered by structures determined from the transversalis fascia, from the internal oblique muscle, the external oblique muscle, and the rest. During this process also, as it enters into the abdominal cavity, it will be carrying part of a peritoneal continuation. And this area here is what was a part of the peritoneal here with the fluid-filled cavity that sometimes gets filled up with the fluid. When it is filled up with the fluid, like people with elephantiasis, then you end up with what you call hydrocele, where the testis is filled up with the fluid. That's what we have in this position. The testis itself consists of tubular structures that develop from the germinal cords, sex cords, that form the seminiferous tubule, the rete testis, the straight tubule, and also out of the testis, we have what was developing from the mesonephric tubules, which we refer to as the efferent duct that connects to the mesonephric duct. The mesonephric duct, remember I said, develops into the epididymis that has got a head, a body, and a tail. And then the vas deferens, and then ejaculatory duct. You want to call it the main functional unit, that is where you have the development of the spermatocytes, is the seminiferous tubule. Seminiferous tubule. But we'll be looking at the seminiferous tubule structure shortly. The testes are covered by derivatives from the abdominal wall. We have what we call the spermatic cord that cover the testis. First, we have the scrotum. Remember the scrotum, which is the skin covering the testis at the area of the labioscrotal swelling. So the scrotum consists of the skin. It has a tunica ductus muscle. Tunica ductus muscle. This is a smooth muscle that normally causes the wrinkling of the scrotum. Wrinkling of the scrotum. It's very important in thermoregulation also. It is a very thin, that's covered by very thin skin with a lot of sebaceous glands, a lot of fat, sebaceous glands, a lot of sweat glands within the testis, the scrotum. Now, this many sweat glands, very thin area, the tunica ductus muscle are very important, are elements of thermoregulation to make sure the temperature is below normal body temperature. The spermatic cord will contain structures from the abdominal wall. The external spermatic fascia from the external oblique aponeurosis of the external oblique muscle, the cremasteric muscle, from the internal oblique, and the cremasteric muscle is very important in raising the testis close to the body if it becomes too cold, if the environmental temperature becomes too cold. This is part of thermoregulation. Then we have the internal spermatic fascia from fascia transversalis. And then immediately below the peritoneum, if you remember, we also have some thin fascia that forms the tunica vaginalis and tunica albuginea. So this is what we have as the spermatic cord. Now, within that spermatic cord, we have the blood vessels We have talked about the three fascia. We have nerves, nerve to the cremaster muscle, the sympathetic nerves, the ilioinguinal nerve. We have three arteries, testicular artery, taking blood to the testis, and then the cremasteric artery with the vas deferens. We have three veins, very important. Pambiniform plexus is a network of veins returning blood back to the heart. This pambiniform plexus surrounds the testicular artery, a branch of the aorta, and therefore the heat from the testicular artery is lost to the veins, is lost to the pambiniform plexus. So the pambiniform plexus gains temperature as it goes to supply blood, return blood to the body, and the testis loses, testicular artery loses the heat so that when it reaches the testis, it will be below normal body temperature. Within this area, you can have a direct hernia or indirect hernia because it's within the inguinal canal. Now, the testis comprise of the tubular structures. Generally, the tubular structures comprises of an epithelia comprising of different types of cells we'll be talking about. It will have a lamina propria, a very thin layer of lamina propria, of loose connective tissue. Then it will have a muscular layer. Now the muscular layer increases as you move to the ejaculatory duct. It's very thick in the vas deferens and the ejaculatory duct. Very thin in the seminiferous tubule and the other tubular systems within the testis. That muscle is smooth muscle that provides a peristaltic contraction for the transport of testicular fluid and also, of course, transport of spermatozoa. The testicular system, tubular structures comprises of seminiferous tubule. If you stretch it 250 meters long, and within this we have Sertoli cells, spermatogenic cells, and then in between those we have interstitial cells of Leydig. From seminiferous tubule, we have tubule recti. This is a straight tubule from the seminiferous tubule, lined by simple cuboidal with dense connective tissue and some smooth muscle. This opens into arete testis within the mediastinum. Created testis is a network of tubular structures. Network of tubular structures. These are anastomosing tubules comprising of low cuboidal, simple columnar, simple squamous. Then this opens into the epididymis by vas efferent duct. Efferent duct. We have an efferent duct, and that efferent duct now opens into the epididymis, and then from the epididymis to vas deferens. Now the vas deferens has a very thick muscular cord, very thick muscular cord, because this is very important in ejaculation. The entire muscle contracts so that spermatozoa can be ejected out at high pressure. Then the ejaculatory duct. So what is the structure of the seminiferous tubule? What is the structure of the seminiferous tubule? Seminiferous tubule, I've talked about it in terms of structure. It has got one spermatogenic cells and Sertoli cells. In between the seminiferous tubules, we have the interstitial cells of Leydig. Spermatogenic structures, that is spermatogenic structures, cells, include spermatogonia close to the basement membrane, the primary spermatocytes, secondary spermatocytes, spermatids, and spermatozoa in the lumen. So this is produced, spermatozoa is produced by the process of spermatogenesis. Spermatogonia, which we call the germ cell, undergoes mitosis to form primary spermatocyte. Primary spermatocyte. Spermatogonia first undergoes mitosis to form two cells. A germ cell, which is spermatogonia, and a more differentiated cell, which is spermatogonia B. Spermatogonia A is like a stem cell. Spermatogonia B then develops to form the primary spermatocyte by undergoing the mitotic division. So we have a growth phase and then meiotic division. So spermatogonia B then can undergo first meiotic division to form the primary spermatocyte. Usually, you see a very large cell, that's a primary spermatocyte, after spermatogonia. Then after that, you have secondary spermatocytes, very small. That is after primary spermatocyte undergo meiotic division to form now a secondary spermatocyte. And that secondary spermatocyte undergoes the second meiotic division to form the spermatids. Those spermatids transform to become spermatozoa by the process of spermiogenesis. So spermatogenesis is divided into two processes. Spermatocytogenesis, where you have the formation of the spermatocyte, the spermatid from secondary spermatocyte, then that spermatid undergo morphological changes now to form a spermatozoa. Those morphological changes are what we refer to as spermiogenesis. Comparing these two, we have the spermatogonia The spermatogonia, like I've said, undergoes mitosis, forms spermatogonia A, which is a stem cell, and spermatogonia B, which is a more differentiated cell. Spermatogonia B then undergoes first meiotic division to form, undergo mitosis, to form primary spermatocyte. And then that primary spermatocyte now undergoes first meiotic division to form secondary spermatocyte. And the secondary spermatocyte undergoes second meiotic division to form spermatids. Then spermatids undergo spermiogenesis. So from one spermatogonia B, we end up with the 4 spermatocytes. From the oocyte, similarly, the oogonia, we have the primary oocyte, that's after first meiotic division, the secondary oocyte, and that secondary oocyte will undergo second meiotic division after fertilization, only after fertilization. Oocyte meiosis. This is the seminiferous tubule. Here we have the interstitial cell. This is between the 3 seminiferous tubule. We have interstitial cell, some smooth muscle, 1 or 2 layers of smooth muscle. There's a basement membrane, and this is an epithelia, somatogenic epithelia, specialized type of stratified epithelia. So the spermatogenic epithelia will have spermatogonia very close to the basement membrane. The spermatogonia, like I've said, undergoes mitosis to form the primary spermatocyte. So mitosis to form spermatogonia A and spermatogonia B. Spermatogonia A is just like the spermatogonia. Spermatogonia B now divides to form primary spermatocytes. These are what you see. And primary spermatocyte continue on as secondary spermatocytes. And then finally, in the second meiotic division, you have the spermatids. These are spermatids lodged in Sertoli cells. Spermatids lodged in Sertoli cells. So that's what we have as the spermatogenic epithelia. What we have also among here are Sertoli cells. It's now 10 o'clock, class rep. Do you have a class after this? Class rep, do you have a class after this?
Yes, we have a class at 11.
At 11?
Yes.
Okay, if you have a class at 11, this will allow me to finish male reproductive system. Just allow me by consensus if you would like. We agree we continue to finish male reproductive system?
Yes, you can just finish.
Nothing, class rep? Aaron? Aaron is not logged in?
Yes, we can continue.
Okay, fine. So this is what we have as the structure of the seminiferous tubule. Now, Sertoli cell, where you see these spermatids, that's actually, they're lodged in Sertoli cell. Sertoli cell extends from the basement membrane all the way to the lumen. It has several processes. several processes, and these processes connect to each other, forming junctional complexes. These junctional complexes, which attach and detach, divide this epithelial lining into different compartments. So the spermatogonia and the primary spermatocyte and spermatogonia B form what you call adluminal compartment. So that's what you call adluminal compartment. The more mature developing spermatogenic cells, that's the secondary spermatocytes and the spermatids and the spermatozoa, form the luminal compartment, very close to the lumen. The other one is adluminal compartment, close to the basement membrane. This is very important in dividing the different epithelial, by forming epithelial junctions, dividing this wall into different functional compartments. The requirement of the development of the primary spermatocytes, the secondary spermatocytes, the spermatids are completely different. And therefore, we need this type of an arrangement. 2, this is an epithelia. And by definition, one of the characteristics of epithelia is that it is avascular. All the nutrients originate from the blood vessels below the basement membrane in the interstitial space. Those nutrients are absorbed through the Sertoli cell to supply the developing spermatogenic cells. So the Sertoli cell acts like a gatekeeper for the nourishment, protection, physical protection of the developing spermatogenic cells. That's why it creates these microenvironments. But first, I hope you have understood that spermatogonia, from one original spermatogonia, will produce millions